and query building with MedDRA. My name is Suzanne Insur Shaker or Shaker. Both pronunciations are correct. I will be your instructor for this webinar. And I'm a medical officer with the MedDRA MSSO based out of New Jersey. And joining me today is my colleague, Hannah Eaton. Hannah is an MSSO training coordinator, and she will kindly facilitate today's session. So I do like to start with getting to know you a little bit more via the polling tool. So we use Poll EV or Poll Everywhere. You can access the tool at this time, either via the QR code or the URL on your screen. Now, the tool is anonymized and will not identify any users. All responses will be anonymous. But if it does ask you for a username, just use MedDRA123 or skip that part. So let's start with just getting a bit more detail on where you're calling from today. So if you can access the tool and put in the pins, and as we wait for more pins, just a few housekeeping items. As Hannah mentioned earlier, today's webinar is scheduled for 2 hours, and I usually do use the 2 hours because there's a lot of information to go through. And this will be an introduction to data analysis and query building with MedDRA. On April 30th, in 2 weeks' time, we have a webinar dedicated to standardized MedDRA queries or SMQs, and many folks find it helpful to attend that webinar after this one. So if you're interested in SMQs, please do join us again in 2 weeks' time. We are planning on opening up a Q&A session at the end, but if you have questions as we go through the slides, please post them in the chat pane and Hannah will present them to me. So it's always wonderful to see pins from different parts of the world. I think it's a nice testament to MedDRA being an international standard. I would like to welcome everyone joining us, as we appreciate you taking the time in your day, especially if it's maybe a bit early for you or a bit late for you. I do thank you for joining us. For the interest of time, I will keep going, but I'm very happy to see that folks can access the tool and the pins are showing. Okay, a second question. What role or roles or functions do you currently perform, may have previously performed, whether in this life, in previous lives? And I do like seeing this slide. It's one of my favorites because in some webinars, we end up with attendees who cross every single possible option. And it's a nice reflection that MedDRA is a tool available to all of you to leverage in your functions across the various operational teams. So again, this is one of those webinars, I think, where we may have representatives from almost every option that we have on the screen. So thank you for joining. Again, the majority, as typical with our webinars, are from pharmacovigilance. So thank you, everyone. And last question is, how experienced are you with MedDRA? Whether you're new to MedDRA or seasoned in MedDRA, I think you hopefully will come away having learned something useful. And again, various expertise across the board. So welcome everyone. All right, so MedDRA was developed under the auspices of the ICH. The activities of my team, the MSSO team, are overseen by the ICH MedDRA Steering Committee, which in turn is composed of different members from both sides of the aisle. So we have regulators as well as industry members across the different ICH regions, as well as observers like WHO, and a couple of new folks as observers as well. And one of the things I like to show when I talk about MedDRA is the MedDRA website, because on the top right-hand corner of it, you'll see the ICH logo. And when you click on the ICH logo, it takes you to the homepage, to the ICH homepage. Under the ICH work products, I'm sure all of you on the call are familiar with these guidelines, the quality, the safety, the efficacy guidelines. But there's a fourth type of multidisciplinary guideline category. And if you click that, you'll see that MedDRA is the first M1 multidisciplinary guideline. So MedDRA is, in fact, an ICH standard. And the ICH website itself will provide you details on the implementation status of MedDRA, and many of the ICH guidelines themselves will reference MedDRA and different levels of MedDRA that are encouraged to be included when you are reporting. So I thought that was always helpful to start off. And of course, it links you back to the MedDRA webpage. But let me go back to the slides and present you with our disclaimer and copyright notice. Just like MedDRA is owned by the ICH, the slides that we are presenting to you today are also owned by the ICH and copyright protected to the ICH. But with the exception of the MedDRA and the ICH logos, you are welcome to use the information provided as long as you acknowledge the copyright and as long as you clearly demarcate any changes you make to this presentation without any inference that these changes were in any way endorsed or sponsored by the ICH. The second bullet point is our disclaimer that the presentation is provided as is without warranty. The third doesn't really apply because we don't have third-party material today. In today's course, we will discuss a document titled MedDRA Data Retrieval and Presentation Points to Consider. This is the ICH endorsed guide for use of MedDRA for data retrieval and presentation purposes. We will discuss a few applications of MedDRA in data retrieval and give examples on presentation and analysis with MedDRA, and maybe have a few demos. I will try to give as many demos as I can squeeze in for the use of MedDRA for these purposes and how to develop queries using MedDRA, and conclude with a Q&A session. So let's start off with the points to consider document. ICH provides ICH endorsed guides for MedDRA users. They call them the points to consider guides, things to think about when you're using MedDRA. They are helpful options when using MedDRA for data retrieval and analysis, whether it's for industry purposes or regulatory purposes. And the best way to apply these documents is in conjunction with each other. So there's another ICH endorsed guide on coding with MedDRA, and that one is called MedDRA Term Selection Points to Consider. Because when you're coding with MedDRA, what you're doing is simply selecting a lowest level term that already exists in MedDRA that is the closest match to the original verbatim or the original natural language term that you're trying to code to MedDRA. These ICH endorsed guides are recommended to be the basis you use when you create your own organization-specific conventions, whether for coding or for data retrieval with MedDRA. Okay, and then these ICH endorsed guides were developed by a working group of the ICH Management Committee, so they do reflect the input from representatives from the industry as well as representatives from regulatory bodies and regulatory members. The update for these documents occurs annually in March, along with the March release of MedDRA, and they are available in 6 languages as complete versions and an additional 9 languages as condensed versions. And they are available on the MedDRA and the JMO websites. And JMO stands for Japanese Maintenance Organization, which is the sister organization to the MSSO, but in charge of MedDRA in Japanese. So in today's webinar, we'll cover some of the topics addressed by this PTC document, especially general principles that will come across in the upcoming slides, as well as a general overview of queries and data retrieval. In our next webinar on 30th of April, we will be covering the topics of SMQs and customized queries. So first, general principle in this document is quality of source data. I think this is something that's known to everybody in this industry, that when you aim for high quality data output, you need to start at the source of information and ensure that your processes for data capture optimizes the quality of the information originally reported. But don't forget, when you're thinking of data retrieval and analysis, your source for that is going to be your MedDRA coded data. So again, you need a good process for coding to optimize coding quality and consistency with appropriate term selection. Organizations are strongly encouraged to pursue continuous oversight of data quality. Data quality issues are, of course, addressed in the points to consider document in more detail. In terms of data conversion considerations, sometimes your source data may exist in terminology other than MedDRA. So for example, whoart, co-start, like when you have legacy data, or when you're performing cross-analysis with older data, or when you've maybe purchased an outside product, or maybe even purchased an outside company. If you want to convert this legacy data into MedDRA, you want to give special consideration to the method used to convert the data, because this can impact retrieval and presentation strategies. So you have two options. You can either convert the data to MedDRA from the original reported term, your verbatim terms. This is assuming you have access to the original reported terms. The second option is you can convert the data from the legacy terminology itself to MedDRA, and results will reflect the specificity of the previous terminology, which may not be as granular as MedDRA, and in which case you're going to lose some specificity that MedDRA offers. So an example, in your original verbatim, the reported term, let's say, was gastrointestinal ischemia. And the legacy terminology that was used did not have that level of granularity. So it was coded to gastrointestinal disorder. Now, MedDRA does have gastrointestinal ischemia, but if you don't have access to the original verbatim, you already lost that specificity. So if data had been converted directly from legacy terminology terms to MedDRA terms, then it could impact how much specificity you can take back out of your coded data. And of course, if newly acquired data are coded directly from reported terms to MedDRA, that's another thing to consider, for resulting differences in specificity could make interpretation difficult. Now, remember that documentation is also very important. It's important to document the data conversion method used, including the date of the conversion and the MedDRA version used. And this ties back into a general second principle, which talks about the importance of documentation. So it's already important to prepare for things like audits, onboarding new team members, ensuring knowledge transfer and continuity in case of changes to the team, and so on. So organizations are encouraged to ensure organization-specific guidelines be consistent with the ICH endorsed points to consider document, whether this is for coding or for data retrieval and output strategies, and also to document your quality assurance procedures. Organization-specific strategies being consistent with the points to consider will guide you through the importance of documenting the MedDRA version, for example, that you used for the search. What are your search strategy methods? Are they sufficient enough in detail to be reproducible? How are you approaching versioning? What is your version update processes? What are your processes for creating and maintaining any customized MedDRA query? Okay, so because in the next few sections of this presentation, we're going to be talking a lot about different acronyms in MedDRA, I just wanted to make sure that folks are all kind of aware what these acronyms stand for. So let's go through a couple of different questions. Let's start with the order of MedDRA hierarchy. MedDRA has 5 tiers. So MedDRA is built in a hierarchical structure. It has 5 levels. What are they in order? So let's give a few minutes and see what folks on the line think. Okay, so maybe we can show the responses at this stage. I know we have more folks on the line, but just for purposes of time, we're going to keep going. So let's show responses. All right, so the correct answer is the one that the majority have picked. So it does start with the LLT, that's followed by the preferred term or the PT, then the HLT, HLGT, and ends with the system organ class. Let's do another question. When you're coding in MedDRA, Data coding is performed typically at which MedDRA level? So this is the level that you interact with MedDRA when you are using MedDRA for coding purposes. Okay, and let's show what folks think. This is interesting. So the majority have picked the LLT, and a good number of people have picked the PT. And we're going to touch base on this a little bit in the coming slides. So this is interesting to see. How about this question? Every PT in MedDRA is linked to at least one primary system organ class and at least one secondary system organ class. Do you think this is true? Do you think this is false? Or maybe you're not sure? All right, so let's see the responses here. Wow, almost half. Okay, very interesting. All right, we're going to address all of these questions, but let's do the last one, which is this question. The primary system organ class assignment or allocation is performed at which stage? Do you select it when you're coding? Do you leave it for your safety reviewer when they're QCing the coding? Do you think it's predefined in MedDRA when you receive the MedDRA files? Do you think the regulator selects it when you submit your data to them? Or maybe you're not sure. So I'm just going to wait a couple more seconds to get a few more responses. Maybe we can show responses now. Okay, the majority think this is predefined in MedDRA, and the majority are correct. But let's give a quick overview, because I think this is helpful to kind of get a feel for what's coming next in this particular webinar. So we said MedDRA is built as a hierarchy system. It has 5 levels that you can use, either from a top-down approach or a bottom-up approach, and they are linked vertically. So each level from the bottom is subordinate to the linked term in the level above it. And of course, those on top are superordinate to the ones linked below them. When you're interacting with MedDRA for purposes of coding, the level that you're using MedDRA is the lowest level term, the LLT. This is the most granular level of MedDRA. It has the most number of terms. And as of version 28, we had close to 90,000. We're at version 29 now, so there's a few more than that. So it has tens of thousands of LLTs that you can search for, and that reduces the need for interpretation at the time of coding, because you can look through them and try to find one that accurately records the reporter's words. So the LLT terms are meant to portray different ways of conveying a condition or a medical concept as close as possible to the natural language that you would receive in your report and as practiced in real life by the reporter. Many LLTs end up being sibling LLTs. They are very close to each other, and they just represent different ways of saying the same medical concept in different words. So they could be semantically equivalent LLTs, they could be synonyms. So they really portray the same concept, but are worded a little bit differently. So you could say, for example, hearing loss, or you could say deafness. They're both siblings, they represent the same medical concept. When we have sibling LLTs, we only need one to represent this medical concept at the preferred term level. So in this example, there's an LLT of deafness and an LLT of hearing loss. Out of these two, the one that we prefer at the preferred term level is a preferred term of deafness. So in MedDRA, the term deafness will be found not only at the LLT level, but it's also found at the PT level. That LLT is just preferred to the rest to represent this standalone unique medical concept. So basically in MedDRA, every preferred term is duplicated at the lowest level term. Same text, same exact numeric ID. So in the question, when we asked you, what level do you use for coding? The answer is LLTs, but folks who also checked PTs are not technically wrong, because each PT is also repeated at the LLT level. But notice the difference in total number of terms as you go higher up in hierarchy. We see that the LLTs that were almost 90,000 got constricted into about 27,000 preferred terms. This is because many of the LLTs, like we said, are siblings and they represent different ways of saying the same medical concept. They might have extra details at the LLT level that may be useful for the LLT level, for that level of detail. But MedDRA starts aggregating even at the PT level, and some of these details will just disperse a signal if they are at a PT level. Now, each LLT is only connected to one PT. So it goes from LLT to PT in one connection. As you go higher up the hierarchy beyond the preferred term level, none of these levels above the PT are used for coding, because now you are in the realm of data retrieval and data analysis, because PTs, or preferred terms, that are related to each other medically in MedDRA get grouped together in the next level at the HLT, or high level term. This is a grouping term. It's intended to be medically meaningful, so it can be based on anatomy or pathology or physiology or function. The folks who decide this grouping are MSSO team members called IMOs, International Medical Officers. They are physicians. They are experienced not only in MedDRA, but also in the use of MedDRA in safety analysis. And look how the numbers have constricted even more. Now we're under 2,000 at the HLT level. This number becomes even more constricted at the next grouping level, because HLTs that are related to each other are also grouped again in clinically relevant ways. And finally, you reach the HLTs themselves get grouped into the final hierarchy or level in MedDRA, which is the system organ class. We only have 27 of these system organ classes, and they're usually grouped by etiology, site of manifestation, or purpose. Now, the hierarchical structure of MedDRA was intended to facilitate data retrieval by providing these grouping terms, the HLTs and the HLGTs. They work to aggregate very specific terms used for coding into broader medical categories. And just looking at the numbers from the bottom to the top, it shows you how signals can get condensed, starting from the natural language verbatim all the way to the system organ class. In many organizations, data summarization, and data presentation, and data analysis starts at the preferred term level. So the PT level is very important. It's also typically used in reporting and submissions to regulators. So if you go back to the ICH guidelines in safety and efficacy, you're going to see preferred term levels referenced in some guidelines like DSUR PrEP and the periodic benefit risk evaluation PrEP, the PBRA. Now, we said that each LLT links only to a single preferred term, and every preferred term has a link that eventually takes it to at least one system organ class. But it's very important to know that there are many preferred terms, just under half of them, in fact, which can be linked to more than one system organ class if that applies to the medical concept. So think about a term like skin cancer. It's a cancer condition, so it needs to go to the neoplasm system organ class. But it's skin cancer, so it's also a skin condition. So it needs to go to the skin system organ class, which means that skin cancer in MedDRA is linked to two system organ classes. When you run a default output of cumulative data coded to MedDRA, you really only want to count each preferred term once. You don't account this one instance of skin cancer as two adverse events, right? So that means that out of these two system organ classes, one of them needs to default as your primary in your cumulative data listings and be the one that defaults when you're presenting your data. So this choice of which one of these two is the primary is again made by the IMOs, by the medical officers. So it's defaulted into MedDRA. It's hard-coded into the MedDRA files that are provided to you. All other system organ classes that are linked to that preferred term are considered secondary system organ classes. Now, how do we decide which one of the system organ classes is primary? Usually is based on the site of manifestation. The primary path that an LLT takes to get to the primary system organ class is usually the system organ class that represents the primary site of manifestation. So if you have a medical concept like a gastrointestinal concept, like a gastrointestinal ulcer, this would go primarily as a primary link to the gastrointestinal sock. If you have a respiratory concept like a cough, it would go in a primary link to the respiratory sock. And this is the predefined path from the LLT to the primary system organ class. Now I'm going to show you the list of 27 system organ classes, just so you can see what they look like. Most of them represent body systems or body organs. So you're going to see the cardiac sock, the endocrine sock, the eye sock, the ear sock, and so on. There are three important system organ classes that represent significant safety signals. And I'm going to show you what they are, starting with infections. So there's infections and infestations. There's the congenital, familial, and genetic disorders, and there's neoplasms. Because one of the important things to know about your product or device is whether or not it could be associated with congenital deformities, those under the congenital sock, or with cancers, those under the neoplasm sock, or with infections, those under the infection sock. So these three system organ classes represent an exception to the rule of how we assign the primary path, because these are not sites of primary manifestations. They are etiologies. So remember when we talked about skin cancer? Yes, it is linked to two socks, the skin sock and the neoplasm. But for skin cancer, the primary link, the primary path, the primary system organ class is not the site of manifestations. It's not the skin. It's in fact the neoplasm sock. And why is that? Because we want to force that signal in your default reporting. We want that as a safety signal that is defaulted when you look at your data in their primary sock. It's more important to identify that the product may be associated with cancer, no matter which organ or body system is affected. Same for congenital deformities, same for infections. So this was done to facilitate signal identification, since all PTs relating to such categories are grouped together on routine cumulative data outputs in order to aggregate these critical issues, the congenital anomalies, the neoplasms, and the infections, and facilitate early safety signal detection. Okay, if you're wondering, well, what happens if the same preferred term is linked to more than one of these three critical socks that you just mentioned? So if we have, for example, a preferred term, congenital infection, this is both a congenital condition, it's also an infectious condition. So the decision was made that the one that is most important to prioritize in default reporting is the congenital sock, and that's followed by neoplasms and followed by infections. So for congenital infection, the primary sock would be congenital and the infections would be secondary. When a preferred term, like these congenital infection and the skin cancer, when a preferred term is linked to more than one system organ class, that's what we call multi-axiality in MedDRA. And MedDRA being multi-axial, it allows it flexibility. It allows you decisions and choices and options in your data retrieval, because not only do you have the ability to view your data in the primary sock, you also have the ability to look at your secondary paths in MedDRA. So again, let's think about skin cancer. Let's say that you understand why it needs to be linked primarily to the neoplasm, because if other patients or other cases had neoplasm anywhere else, you want them all to be aggregated and grouped together and you want to enhance that signal. Fair enough. But in your particular scenario, you're working with a topical product, and this topical product is causing different types of skin problems. Skin cancer is only one of them. And you want to be able to count all the probable skin manifestations that are happening with the use of your topical product. So you want to see the skin cancer, but you want to see it under the skin sock, and you want to be able to count that adverse event with the remaining ones. That's when we can leverage the secondary link to the skin sock. And we're going to talk a bit more about this in coming slides. And one final thing to point out before we move on from this slide is that there are three kind of special socks in MedDRA called uniaxial. And uniaxial socks basically means that the preferred terms that exist in these system organ classes only exist in these system organ classes. They're not linked to any other system organ class in MedDRA. When you think about them, it's going to make a bit more sense. The first one is this one, investigations. So investigations like labs, x-rays, scans, they could be related to so many various conditions, multiple body organs, different body systems. Same with surgical and medical procedures. So really, it's not really feasible to link them to every single possible SOC they could be related to. It becomes overly complicated and not really useful. Same for the third one, which is social circumstances. So social circumstances, you can think of them as kind of risk factors. Somebody who is a smoker, somebody who has traveled internationally. These are folks who are at risk for many different conditions. It won't be accurate to limit which SOCs you think that can be linked to. So a decision was made that these three special kind of SOCs are uniaxial. One thing really important to remember about these three SOCs, because they are uniaxial, you need to be very intentional about looking at them when you're building queries in MedDRA because of the fact that they don't have multi-axiality that you can leverage. Okay, so I'm going to move on and talk about the general principle number 2. Don't change MedDRA. We just said that MedDRA is hardwired with the primary SOC allocation. So it's a standardized terminology. It has a predefined hierarchy. It should not be altered. Don't make any ad hoc structural alterations, even if you're able to, because that would compromise the integrity of this standard. And please be aware that if you're working with other partners, other colleagues from different companies, if you're submitting your data to regulatory authorities, all of these folks are using MedDRA in its standard terminology. So if you choose to change the structure of MedDRA, you are no longer using this standard and making it more difficult for folks like a regulatory authority agent to compare the safety data of similar products from two different organizations if one of them changed the primary SOC allocation based on maybe a recommendation from one of their safety physicians. So just please be careful not to make changes to MedDRA. However, if you suspect that improvements can be done to the structure of MedDRA because you think that one PT is linked to a primary SOC when another one is actually better, then please feel free to submit a change request to the MSSO. There are links at the end of these slides showing you how to get to that tool, and each one of those requests is fully reviewed by the medical officers. Okay, another general principle talks about how organization-specific characteristics can impact how you use MedDRA. So although MedDRA is a standardized terminology, different organizations can implement it in various ways. It's important to understand these organization-specific data characteristics because they're going to affect your implementation strategy. They will determine which levels of MedDRA are easily available to you. What tools we will show you today are available to you and can be leveraged within your databases. So it's useful for you when you're making some decisions on how to build the database is to have someone at the table who is a MedDRA specialist, because they can give you input on issues like how is the database structure going to affect the MedDRA hierarchy that is stored and that is used? Which level of term or reported term are you storing in your data? Are you using synonym lists? And if so, are these at a global level or study level? What is your data conversion methodology, if that applies to you? What are your coding practices and how do you change them over time? And of course, don't make assumptions, because there could be limitations, restrictions you're not aware of because you made assumptions. Don't assume, excuse me, that you're going to have access to the secondary SOC locations. You may not have that access, or it may require a special request. And of course, for term selection principles, one of the questions that comes up is, does your organization have a one-to-one correlation of verbatim to MedDRA code? Or are you allowed to select as many LLTs as apply? Now, for example, on the AE reports, if you receive a diagnosis with a sign or symptom, are you only selecting an LLT for the diagnosis, or are you coding the diagnosis as well as the signs and symptoms? Because it's going to affect the counts of terms, which will impact your AE profile. And then of course, the adverse event profile that results when you're using both diagnosis and signs and symptoms may appear different than that used when only the diagnosis is coded. So these are kind of coding conventions that you have to think about early on when you're building, but also it's going to affect how you compare data from other databases if you're co-developing or co-marketing with partners or submitting to regulatory authorities. So a few things to think about. Now, there are MedDRA characteristics that can also impact data retrieval and analysis. We talked about the HLT and the HLGT levels, these grouping terms. They are a great tool for data analysis and retrieval. I don't think they are used as often as they should be, because they're really very helpful, and they can provide you with clinically relevant groupings of terms. So let's say that, for example, in one of your particular products, you're interested in cardiac arrhythmias. Well, there's an HLGT, a high-level group term level in MedDRA, that is specific to cardiac arrhythmias. And within that HLGT, there are subgroupings of specific types of arrhythmias. So if you're interested in one particular type of arrhythmia, let's say supraventricular arrhythmia, then rather than retrieve all the patients within your cardiac disorder SOC to review them, or even all the cases within the HLGT cardiac arrhythmia, you can even restrict your review to the cases that exist in PTs that are subsumed under that specific HLT for the supraventricular tachycardia. But one thing to point out is that before you decide that, okay, a grouping term in MedDRA makes sense for me to focus on when I'm retrieving cases, please look at all the PTs in that group before you decide you're going to use it. Because not every group may fit the purpose that you're hoping for. This point is driven home when we look at an example from the investigation SOC. So one of your products may be associated with a decrease in blood pressure. And you look in MedDRA and you see that under the investigation SOC, there is a grouping term, an HLT level term called vascular tests, not elsewhere classified. That's what NEC stands for, including blood pressure. And then you think, okay, maybe this is a good one for me to interrogate my data and retrieve all my relevant cases of blood pressure decreased. But if you look through this HLT with all the PTs that are subsumed under it, you're going to find that it not only includes blood pressure decreased, but also it has blood pressure increased. In fact, it also includes blood pressure abnormal, blood pressure normal, and the fact that they just measure the blood pressure. So it has blood pressure measurement. So it is grouping all of these blood pressure investigations together, yes, but whether it's high, low, normal, or abnormal, they're all in the same HLT. So in this case, it may not be a good choice for you if you want to be specific in targeting cases just for blood pressure decrease. You're going to end up with a lot of noise if you use the entire HLT. Same with a few clinically related PTs. So sometimes related preferred terms might be overlooked because they might be in different groupings in MedDRA. They could be in different groupings within the same SOC, like in different HLTs or different HLGTs, or they may even be located in more than one system organ class. For example, in the skin SOC, there is an HLGT, a high-level group term that contains dermal and epidermal conditions. And this HLGT has subgroupings. One of them is bullous conditions, and one of them is exfoliative conditions. Now, if you're interested in a condition like Stevens-Johnson syndrome, and you only look for cases using the HLT bullous conditions, you're going to miss relevant cases where the reporter used words like skin exfoliation or exfoliative dermatitis. And that means that if you ignore that HLT, the frequency of that medical concept will be underestimated because you're not capturing everything, and that would impact your interpretation of data. So the takeaway is please spend time when you're building your queries. Look through MedDRA. It's worth the time, it's worth the effort, it's worth the consultation of different team members who understand the product safety profiles, who have maybe a medical background, and who understand MedDRA. Now, we already touched base about what we mean by multi-axiality. So it is when a PT exists in more than one system organ class, and this allows terms to be grouped in different but medically appropriate ways, like etiology, for example. Now, each PT is assigned one primary SOC. And like we said, some PTs are linked to more than one SOC, in which case all other SOC assignments for that PT are called secondary. Having a single primary SOC prevents double counting of events, like we said. So when you output your data from SOCs, you are limiting it to one record per preferred term. But if you don't look at your secondary SOCs during your analysis process, you may not be getting the accurate frequency of the medical condition of interest. Remember that skin cancer? If you're only looking at the data default, that skin cancer is only going to show up under the neoplasm SOC. But if you're interested in skin manifestations and you want to count it, you got to leverage the secondary SOC output. All right. And it's nice to remember that PTs relevant to a specific medical condition of interest may exist in MedDRA in multiple SOCs. So if you're interested in renal abnormalities, you can certainly start your search in the renal SOC. That makes perfect sense, but it's not enough. You must remember to look in those three special SOCs, like we said, that are uniaxial. You have to look in investigations, like you're going to find lab tests related to renal disorders. You have to look in the surgical SOC, where you're going to find procedures related to renal disorders. And don't forget social circumstances, where you're going to find risk factors that may be associated with renal disorders. So if you fail to consider data coding in the non-multiaxial SOCs, you're going to have an incomplete analysis of renal abnormality. Okay, we already said how data presentation defaulted to the primary SOC is a good starting point when looking at your data, especially to quickly identify critical issues, especially congenital conditions, neoplasms, infections. In fact, usually this is typically the default presentation when reporting. But remember that looking at your data using secondary SOCs in your data presentation will give you a different view and a more accurate picture of event frequencies. And as a note, regulatory agencies that you submit your data to are very familiar with this concept in MedDRA. And so they are familiar with these alternate views and will often perform that analysis on your submitted data. So let's see a practical example of what we talked about so far. Let's say you ran your cumulative data listed and sorted by primary SOC, and your team came back to you and said, we suspect there's a potential issue with the investigation SOC because the frequency of events reported was significantly higher for the group using this experimental product, my drug, when you compare it with the placebo group. And this is from initial analysis. And you were tasked to look at the output and identify whether you see a trend. So you start looking and you have only your preferred terms. So you're trying to determine whether you find something of interest here. And you may already be suspicious of something because you see higher levels of AST increased. you have ALT increased, you have GGT increased. So you're starting to think, well, maybe it's the liver. The remaining standalone PTs may not be very significant. And you go through the first page and say, okay, I think I'm good. And you go through the second page and you go, okay, I don't see a lot of other stuff here to worry about. But you're only looking at groups of preferred terms, individually listed in your listing. So let's say that you attended this webinar. You remember that we said MedDRA already does a grouping for you simply by the hierarchy. So you go back to your data manager or your programmer and you say, why don't you run me the same output, but this time do it at the HLT level, one level higher than the PT. Of course, depending on whether or not during your data build, you were able to capture that level. And they come back to you with that same data output, but this time at the HLT level. This is what it's going to look like. And looking at the numbers, it's going to be so much easier now and clearer where the trend is. You can say with confidence now that there's definitely an issue with the liver function analysis. And depending on your internal guidances, how familiar you are with the safety profile of your product, you may even decide to take a closer look at tissue enzyme analysis or digestive enzymes or white blood cell analysis. But now you have an easier way to cut through the numbers, no matter how many events you have, because this example maybe only had 2 pages worth of data. But what happens when you have hundreds of pages worth of data? And I do hope that some of you are curious enough now to go back and actually test it out and request data from your data manager or programmer, sorted first by SOC and PTs, and next by HLTs, and even by HLGTs, and take a look. Okay, another general principle is about MedDRA versioning. MedDRA is updated twice per year. The version that ends with X.0 comes out every March. It contains both simple and complex changes, which means that any level in MedDRA could potentially be affected. Version X.1 comes out every September, and it contains only simple changes, and that's at the LLT and PT levels. When you're planning or performing data retrieval and presentation, one of the very important bits of information to document is which MedDRA version was used. So it's important for organizations to be aware of the types of MedDRA changes in each release for their possible impact on data output. To that end, the MSSO provides resources to assist MedDRA users. The MSSO provides a document called Best Practices Document. You can refer to that one on the MedDRA website. This provides you information on versioning options for clinical trial and post-marketing data. The MSSO also provides a document with each release called the What's New document. It outlines the changes for that release. When you receive the release packet every 6 months, it includes a version report that specifies changes associated with that release. It's basically a spreadsheet. It lists all changes between the current version of MedDRA and the version that just preceded it. But the MSSO also gives you a tool. It's included with your current subscription. It's free to MedDRA users, and it's available to everybody on the call. And this tool is called MedDRA Version Analysis Tool. And it facilitates identification and understanding of the impact of changes between any 2 MedDRA versions, not necessarily consecutive ones. So another important takeaway from this webinar is when terms are used for queries, the version of that query must be in the same version as the data being queried. Otherwise, your output is not going to be very accurate. So let's give you examples about the changes in version and how that can affect your data review and data analysis. Okay, in MedDRA version 22.1, MedDRA had separate terms at the preferred term level for pelvic fracture and ischium fracture. Now, based on MedDRA end user change request, a change request was submitted and said, the term for ischium fracture really belongs under pelvic because it's part of the pelvic bones. And the IMOs agreed, and so the preferred term, ischium fracture, was demoted to an LLT level, and it was placed under the preferred term of pelvic bones. Pelvic fracture, sorry. So now let's say you had used the preferred term ischium fracture in a query on your data in version 22.1. And at that time, you had discovered 15 cases of that condition. If the query had been rerun on data using MedDRA version 23, these events are not going to be found at the PT level because the PT fractured ischium by then had been already demoted. So that's very important to know that they didn't disappear. They will appear under a different preferred term. Here's another example. Same version, 22.1. The preferred term vascular cognitive impairment had a primary link to the psychiatric disorder and secondary links to the nervous system and the vascular disorders. In version 23, the primary SOC assignment was changed to the nervous system disorder. They kept the psychiatric SOC, but it now became a secondary link. So in a primary SOC output of data, if you're looking at your data defaulted to the primary SOC, you're going to think these 20 cases of vascular cognitive impairment just disappeared, but they didn't disappear. They moved under a different SOC. So it is important for you to learn about the MedDRA version analysis and how to leverage that tool. We're going to go a little bit over the tool and give you a quick demo. Okay, so the tool is available in more than one spot. We have it on the website. I'm going to show you where. We also have released, the MSSO has also released a new version of the web-based browser with the tool embedded in it. So the current or legacy version of the web-based browser does not have the MVAT tool, but the new browser has it embedded within it. Of course, it's free, it's part of your subscription, and it has three main functionalities. So you can compare any two versions of MedDRA and have an exportable report outputted that you can give to your programmer and say, here, check this against our data and see if any of our PTs that we used have been impacted between these two versions. You can actually import your own data that you coded to MedDRA. So it's MedDRA coded data, but that's your data. You can import it into the tool and select in the tool which version these data were coded to, and ask the tool what changed in my data or what would impact my data against a different MedDRA version. And you can look at a single term in the tool and follow that term from when it was added to MedDRA up until the current history of that term. The nice thing about that tool, just like the browser, you can change the interface language to any language you're more comfortable with. You can also use it for different MedDRA languages. So it allows that. It also allows you to compare data on supplemental changes. And supplemental basically means that these changes have been reviewed by the medical officers. They have been approved by the medical officers. They're not yet officially released. So they're sitting waiting for what the next version release will be. So if we have now supplemental changes, they're going to be officially released in September. But the IMOs have already reviewed them and approved them. This tool also allows you to run your reports using and including the secondary SOC changes. There is a nice place online that I'm going to show you where there's a demo for this tool. I'm going to show you a demo now, but also want to show you where to go to get that tool. So a couple of things before I do the demo about the website. So we're back to the MedDRA.org website. I want to show you where that points to consider document is, and then where the training material is for the MVAT tool. So let's start with the document. We talked a lot about this points to consider document. We said it's available. So where can I find it? So there's more than one way, and that's typical of MSSO tools. There's more than one way to find them. One way is to go under How to Use. How to Use is a very nice tab for you as MedDRA users. So How to Use is a nice one as a user. You go down to Support Documentation. So if you're looking for a document, go to Support Documentation. When you click on it, you're going to see different color banners. There's a blue banner, and there's a lot of gray banners. The blue banner includes all the documents that are ICH endorsed. So you have the ICH endorsed guideline for MedDRA term selection, which we said is the coding. There's the ICH endorsed guideline for data retrieval and presentation, and they're available in different formats to make it easier for you. They also have them in red line, so you can see what changed from, if anything, from the previous version. And sometimes it's only the version or the date that's the change, and sometimes there are a few other changes. So you have the red line. And there's a document, a living continuous document called a companion document. This is of interest to folks who have a interest in specific coding topics, like, for example, medication errors or product issues. Here's the best practices document. This is the document we said that walks you through the versioning options. So you have all of these details in it about how to approach version control and what are your recommendations or options for versioning. The gray banners are version specific. The gray banners come out with every release of MedDRA. And these are produced by the MSSO. So here's your What's New document. It walks you through the changes, what changed between 28.1 and 29. Here's your introductory guide to MedDRA. This is a very nice document for people who are coders. So if you're new to MedDRA, even if you're a seasoned coder, take a look at this document. Really interesting, especially the first few pages of it, as it walks you through how MedDRA was created and what are the thought processes in MedDRA creation. Here's an introductory guide for SMQs, and this is something that will be discussed in more detail in the next webinar. And this is a technical document for the distribution file format. The other way to reach the same spot for these documents, there are quick links up here on the top. So one of the quick links is called PTC, so points to consider, things to think about. If you click it, it takes you to the exact same spot, but the blue banner is defaulted open. So that's the first thing I wanted to show you, how to get that document. The second thing is I'm going to demo the MVAT, but before I do that, I wanted to show you where there's a nice recording of the demo that you can go back to if you want to test out the tool at your leisure. So under training, we have instructional videos and we have training materials. I think it's the instructional no, it's training materials, sorry. So under training materials, if you click tools and click down and go down, you're going to see the MVAT. And there's a nice English Part 1 and English Part 2 in detail if you want to kind of play the video, pause it, experiment with the tool as you're watching it, and then play it back and stop and go as you like. All right, let's demo the actual tool. Let me go back to the home page and show you where the tool can be found. So you are a user, so you're going to go to How to Use, and under How to Use, here's the link for all the tools. And when you click on that, you're going to see different tools available to you. You are interested in MVAT at this point, so you click the MedDRA version analysis tool. It gives you a bit of summary about the tool, and here's the link to open it. There's a link to the training materials, by the way, here as well. So let's open the MedDRA version analysis. And by the way, you have been provided temporary login credentials in the chat feature of this webinar if you do want to log in, or you can use your organization specific credentials to log in. And there's some folks who may not be aware that they are able to get those credentials. If you have an email that belongs to your organization's domain, and that domain is what your organization subscribed to MedDRA under, you should be able to get. So let me just show you how to get that. Again, with the quick links on top, there's something called an SSA, self-service application. If you click on that and you're interested to receive your organization-specific credentials from MedDRA, you can request to retrieve either the MedDRA ID or the MedDRA password or both. So if you click on either of them, it's just going to ask you one question: what's your email? And if it can verify that that email certainly belongs to the same domain, they're going to send you the next action directly to your mailbox. Same with the password. But I digress. Let me go back to the MVAT. Okay, so here's the temporary credentials, which are unfortunately only good through tomorrow. You can log into the MVAT tool. And the landing page will show you that you have a few options to make. Is your data in English or something else? I'm going to keep it English. What is your starting version that you're starting with and what is your ending version. And it doesn't have to be consecutive. You can go with the starting version as old as you like. Let's, for example, say 27. But if I choose an ending version that's older than that, it's just not going to work. It doesn't make sense because the ending version has to be one that's more recent. So let me go back to 29. And you can also change your interface language. So if I'm more comfortable with French, I can certainly operate with a French interface if I'd like to. Another thing that I can do is rather than select two different versions, maybe I have my data in 29 and I just want to see what's coming next. So I click on supplemental. We said supplemental is what's going to be released next. It's already been approved. But look what happens. As soon as I click supplemental, it turns pink to kind of remind you that it's fine. You can look at it, you can prepare for it, but these are not going to be in your MedDRA files just yet because they haven't been officially released. And look at here. It defaults to an ending supplemental version, but you can choose whatever version you want to start with, depending on your data. Okay, I don't really want to do the supplemental. I want to do a typical version to version. So I'm going to go back to release, and I'm going to go from the last version, 28.1, to 29. Notice that there is an option here that's not defaulted for secondary SOC information. So you are able to retrieve secondary SOC information when you run any reports in the MVAC. But for our first demo, I'm going to show you straight up without the secondary SOC. I'm choosing English. I'm okay with comparing between 28.1 and 29, and I'm going to just run it. And I'm going to click run and wait a little bit. Of course, it makes sense that the more versions are apart, the more changes they will exist. So the tool might take longer, but it's pretty much a quick output. You can see on the bottom here, here's your summary and here's your details. You have different number reports, different titles, and different numeric outputs here. So if you're interested in the details of what each one is saying, you have that information available to you under this explanation box. So basically it tells you, I'm explaining to you what each of these reports represents. So the first one, for example, report 10, this is all new LLTs that have been included in MedDRA after version 28.1, and you're going to see them in version 29. And of course, because they are all LLTs, we know that some of them will also be PTs. That makes sense. Some of these LLTs, not all of them, of course, some of these LLTs will also be considered preferred terms. But because we need to differentiate them, there is a special report that also gives you what these new PTs are. But I'm not going to go through all of them. That document is available for you if you want, but just kind of an overview. So there's 611 new LLTs added since 28.1. Out of these, 218 are at the PT level. 20 promotions, which means terms that existed as LLTs in 28.1 are now preferred terms, 40 demotions. So this would be interesting to you because if any of these 40s you already used, they're no longer at the PT level. This could impact your analysis, your presentation. Some LLTs may have moved under different PTs and so on. So let's just give you an example of new LLTs. So if I click on it, I'm going to see them alphabetized, and I'm going to see what the LLT was that was added in 29, and I'm going to see which PT it was linked under and the rest of the hierarchy. So I'm not seeing anything for 28.1, of course, because these are all new to MedDRA. There was nothing in 28.1. What about if I click on promotions? Promotions, we said, they were LLTs that now have become PTs. So yes, there is information of what they looked like in 28.1 and information of what they look like now under 29. But every single term only appears once. And you see the colors in the rows. It goes from white, blue, white, blue, white, blue. It just continues. That's because I didn't choose the secondary SOC to be included in my report. Well, what happens if I do choose the secondary SOC to be included? Let's run this again. But I want to show you one thing. before we run this again, reports 60 and 70. Reports 60 and 70 are changes in the SOC allocation. Since I'm ignoring the secondary SOC in this particular report, it's focusing only on the primary. So I'm going to see 21 LLTs that had changes to their primary SOC allocation. So for example, there's my fever malignant. The primary SOC allocation is the congenital under hyperthermia malignant. And 4 had changes to the primary SOC for PT levels. If I rerun this report, I'm going to go back home and I'm going to keep it 28.129, but I'm going to click this box and I'm going to say, show me the changes, but include the secondary. And I'm going to run it again. So again, it's going to think about it and it's going to output some information at the bottom. Same 611 that were added. That's fine. Same 218. If I look at these 611, I'm going to see that the colors are no longer just always direct, white, blue, white, blue. Here's 2 whites in a row, and the same LLT is repeated. And the reason for that is because this particular LLT links to this particular preferred term, myeloproliferative neoplasm, and this preferred term links to more than one system organ class. So look at that. It's linked to blood and it's linked to neoplasms. And the report will tell me which one of these is the primary SOC because it's hard-coded. Here it is, the neoplasms. Well, why do I have 3 blues in a row? That's because this particular LLT under this preferred term, and this preferred term has 3 system organ classes that is linked to. The primary is renal because it is a renal manifested condition. It's a renal AV fistula. So this shows me that although I still have the same 611 new LLTs, but because I clicked that box for secondary SOCs, I'm seeing all the possible system organ classes that are linked to those terms in MedDRA. But the numbers that did change are 60 and 70 because I no longer see only the 20-some entries here. I'm actually seeing all possible SOC changes, not just the primary. And the same for number 70. I'm seeing all possible SOC changes. So can I use this somehow that's easily exportable, or do I have to copy everything manually? No, you can export. Here's your selection for export. It defaults to everything. So if you're interested in all of these types of changes, you can default everything. One of the most important outputs in this report is currency changes. Those of you on the call who understand or work in MedDRA coding know that you're not supposed to select a non-current LLT when you're coding. Currency changes will show you all the LLTs that changed, either from current to non-current, as we see all of them happen to be that way, or from non-current to current. Now, why is this important? Because if you had chosen any of these LLTs in your coding, in your version 28.1 or even older, now in 29, they are no longer current. So one of the determinations you need to make when you're upversioning is, what do I do with these? Do I recode them to something that's now current? We said you can export them, so I'm going to leave it defaulted to all the options and click export, and it's going to open up an Excel sheet. I'll be able to save that to my folders. I'll be able to customize it. I'll be able to share it. I'll be able to have a discussion with other team members because now it's mine. This output is mine. And it walks you through the same information, all of those reports, each in a separate tab. And it has these delta delta notes with these cells to kind of explain what each of these reports is truly giving you. So I hope that you find this tool helpful. So that's the first thing, the first action that we said. You can compare 2 different MedDRA versions. You can do data impact report. You can upload your own data that you coded to MedDRA. It's not your verbatims. you're uploading your own MedDRA coded data into this tool. So let's say I have data in MedDRA version 28. This is the most important thing before you start using the tool, is know your version. If my data exists in version 28, I have to select the starting version as version 28. Otherwise, my output is not going to be reflective of my data. And I want to see what impact will happen to my data between version 28 and version 29. Let's actually go for 27. I think my data is actually older than that. I think it's at version 27. Now, how am I going to prepare my data to be able to use this tool? And by the way, the tool doesn't save your data. It doesn't save the data. So it allows you to run the tool, to run the report, but you need to export it and save it. Otherwise, it's going to disappear right after you run it. It's not going to hold it. It's not going to keep it. So, okay, I'm interested in comparing my data and my data impact between 27, 29. I don't really want my secondary SOC. I just want to see what impact this version had on it, just from a primary SOC. And I'm going to go next. And it's going to give me a very simple kind of commentary. It's going to tell me what kind of data I need to give it. It says it needs the data in Office Excel, and it needs only 3 columns. If you have a tracker that already has more than these 3 columns, you don't have to remove the information that's extra, but the tool is not going to look at it. It only looks at columns A, B, and C. It assumes that column A is going to be used for identifiers. You don't have to fill it if you don't want to include any row IDs or case IDs, but it's optional. And it's actually useful because it will help you, especially when you have a large amount of data to go through, which row was the relevant one. But if you don't want to put in any information, that's fine. Just keep the column with the column header. It expects the MedDRA term to be in column B. And column B could represent either the LLT or the PT, because some folks keep their data at the LLT level and some folks keep it at the PT level, and it works in either one. And if you don't know what level you have, that's fine. You can just run the tool and the tool will figure it out. Column C depends. It's the numeric code of whatever the information is that you plugged into B. This is the numeric code. If your data is English, you don't need both of them. Either one will be fine. But if your data is any other language other than English, then you definitely have to use the code. Okay, you can either use the code on its own or use both of them. And it reminds you that it's very important, your imported data, you have to know the version of your imported data because that's your MedDRA starting version. In my case, it's version 27. It reminds you it doesn't store your data and that it will be erased after you generate your report. And it notes that the maximum number of records is 100,000. This is the one difference between the new web-based browser that has the embedded tool, because there's no row restriction in that tool. So that's a nice feature. And then, of course, it reminds you that these have to be MedDRA terms. If you import verbatims, those rows that have verbatims that don't have a matching MedDRA term, letter for letter, are not going to be run in the tool. So let me show you an example of what this looks like. Let me open up an Excel sheet and just show you what it's going to look like. Here's my information. My tracker is not that big. It's about what, 200 adverse events. There we go. I have 200 adverse events. I didn't use a case identifier. I just used a row ID that's numeric, just because I didn't want to leave it blank, but I don't really have to fill in these if I don't want to. Here are my either LLTs or PTs. I'm not 100% sure what level it was when it outputted, so I'm just going to leave it without a header, or I'm going to say LLT/PT. I'm not sure what it is. And here's the code. It came out with when the IT colleague sent me this report. I'm ready to use it. I can't keep the Excel open. It won't work if the Excel is open. So I'm going to close the Excel and I'm going to choose my file. So this is my MVAD demo. I went into my folders to find it. I'm going to double click on it and I'm going to click next. Now the tool is processing my data through this data validation. If it finds information in there where I may have made a spelling mistake, or maybe I plugged in one of the rows as the verbatim, but it's not really a MedDRA term, it's going to tell me how many of these rows were not validated. It won't ignore the entire Excel, but just the rows that it didn't validate. In my case, they're all MedDRA terms. So it's telling me that I've uploaded 201 rows. It validates 201 rows. And it tells me what percentage of my data is impacted between version 27 and version 29. So how come there's all of these multiple numbers here? Well, these numbers are not the changes that impact my data. These numbers represent the differences between 27 and 29. But notice it doesn't start at report number 10. There's no new LLTs, there's no new PTs, because basically I'm giving the tool my data coded to MedDRA. I'm not asking the tool to tell me what's new in MedDRA. I've already used MedDRA. So it starts at report number 30. And look at this column, impact on uploaded data. This is the impact on my data. Not a lot of changes happened. Really not a lot. I mean, I see LLTs that moved from one PT to another. Let's click on that and see what happened. Okay, so one of my adverse events was coded to limb salvage therapy. And in version 27, this was under a PT called limb reconstructive surgery. But in version 29, this was changed to limb sparing surgery. And that's the only change in terms of movement between one PT to another. There's no promotions, no demotions. There's no currency changes, which is the really important one. I see 0 there. And I see 4 here under this comprehensive LLT. Comprehensive LLT changes sometimes can be confusing to some MedDRA users because these particular LLTs may or may not exist in my original Excel sheet of 200 terms. But the reason they output here is because they are somehow linked. They may be siblings to other LLTs, or they may be subsumed under a PT that I do have under my report. So some folks don't really use this output for the comprehensive LLT changes because it can be a little bit confusing for them. But the data impact report explanation explains it and tells you that this report really provides you a larger view on all the LLTs that happened that could be related somehow to your list. It doesn't necessarily mean that the LLT was on your list, but it may have moved from a PT on your list or to a PT on your list. And that explains why that particular LLT was not on my list. And I know it might be a little bit confusing, but you'll get the hang of it once you start experimenting with your own data. I think that's a good demo, and maybe I can stop here for a couple of minutes, Hannah, and just ask, are there questions at this point? Because I really want to keep going, but if there are questions, I'm happy to address them.
The only question we've had come in, I think you've already answered, but it might be good to reinforce the point. Someone asked, can you force code to a secondary SOC?
So when you say force code, if your question is, can I change the link that I'm seeing or the SOC that I'm seeing in my default cumulative listing? Can I force it to show me my secondary SOC, like that skin cancer? Can I force it to show me the output as skin rather than neoplasm? You shouldn't, even if you can. I mean, that's the whole point. The point is that MedDRA is standardized. It is linked to both SOCs, and the expectation when you're defaulting just by primary is that you're going to keep the hardwired primary SOCs. Everybody expects that. The regulators expect that. Everybody expects that. But if you're really interested in the secondary and you want to use that for your frequency counts, you can. You can show that in your analysis. You can document that I did this and I'm adding this or I'm adding that. In fact, some guidelines, like if you operate in the EU markets, the EU SMPC has a very interesting section in it that talks about, well, how do we prioritize events of interest? Maybe we want to aggregate things together, even if we are aggregating them not under their primary SOC. So there is general understanding in the industry, especially the regulators, that sometimes frequency counts reflect better accuracy of your safety profile when you're looking at your secondary SOC. But when you are coding, you should never change your primary SOC allocation that's hard coded. So I hope that answers the question, but please come back if you have a follow up. In the meantime, I'm going to keep going because, like I said, there's a lot of information. I will try to give you as many demos as we can squeeze in, and we can continue moving. Okay, now we talked about the first part in MedDRA, which is the different hierarchies and how they can be leveraged for you. Now let's talk about data presentation and how people approach data presentation. And usually the first picture folks usually see of their data in MedDRA is a view by primary system organ class. It really makes sense because primary SOC, like we just said, that's the default setting when you run your cumulative data listing. Most folks on the line, I'm sure, have seen data listings that start with the system organ class, and then they have the preferred terms underneath that. Now, when the MedDRA was created, the original MedDRA expert working group, they realized very quickly that there can't really be a standard alphabetical order of SOCs because MedDRA is multilingual. I mean, we're now up to 27 languages for MedDRA, and I don't know if each single one has their own alphabetical ordering of system organ classes. So there was an agreement to create an international order of system organ classes that, regardless of the language of MedDRA you're using, will be the same. And this, again, enhances and facilitates consistency, irrespective of the language or the alphabet. Now, this international order was based upon kind of relative importance of these SOCs in your adverse reactions or adverse event reports. So alphabetically, the first system organ class in MedDRA that we saw was blood, the blood system organ class. But in the international agreed order, it starts with infections. So it's a little bit different than the alphabetical order, and it is an internationally agreed upon order. So one thing you may want to discuss if you're sharing data outside of your company, understand from your data partners and your regulators what their expectation is. If the expectation is to follow the summary of the international order of the system organ classes, that is made available to you. You do get it in the ASCII files with every release of MedDRA. It is listed in that MedDRA introductory guide that we showed you in that gray banner. It's also mentioned in the summary of product characteristics guideline. So your aim is to present the data in a way that allows for easy recognition of patterns, and sticking to the international agreed order is another way to do that. All right. Something else to consider when you're thinking about data presentation is that the first approach of your SOC and PT approach, this may need in some cases to be augmented with other types of data outputs. We showed you in that investigation SOC example how grouping terms like HLTs and HLGTs can really be of use to you when you're looking at your data. So that's another tool in your arsenal for data presentation. And of course, the methodology of presentations, you can use line listings, you can use tables, you can use graphs, whatever you think is more easier to analyze. Now, representing data just by primary system organ class, whether it is as a line listing or whether it's a table or a graph, will have benefits and limitations. The benefits is that it gives you a quick kind of overview of what your safety profile looks like, especially when you talk about those critical safety signals, your All of your neoplasms will be aggregated in your primary SOC view. All of your congenital disorders, if you have any, will be aggregated together, and all of your infections will be aggregated. So that first look will get that first question done. Do I have any of these significant critical safety signals I need to worry about? So that's your broad overview. Now, the challenge, well, another advantage before I move on to challenges, another advantage is that the PTs, like we said, are only displayed once. So there's no double counting. So your AE profile numeric-wise is representative of your data. Now, there's limitations, and we mentioned these limitations, is that if you don't also take a look at your secondary pathways, you may not be looking at the full groupings. You may not be looking at the full true frequency. طبعا, the item about lengthy output, it depends on your data. If your data is small, most folks are happy with the, with the system organ class, the primary system organ class, and they're okay with that. But if the data is really big, then really your secondary SOC is actually helpful to you because it will give you a clearer idea of your true frequency. All right, let's show examples. It's always easier with examples. Now, in this first output, this is a graphic example of data display by primary SOC. And whether or not you are familiar with your safety profile, whether this is a product that's new to you or known to you, your first instinct right away looking at this graph is that there's an issue happening with the psychiatric SOC. Whether this is because of the mechanism of action of the product, whether it's a known safety issue or a known class effect, whatever the underlying, whether it's a new signal, it's definitely worthwhile taking a deeper look at the system organ class SOC. And I think that's clear to see. And this is another advantage of just looking at the primary SOC. It gives you an issue right away. This example is a line listing example. Now, it's a small output restricted to one SOC, which is the nervous system SOC, but in that SOC is showing all levels of the hierarchy. But because it's a small output, this is actually useful because it's a small output. So all levels of the hierarchy are enough to give me an idea of where the most frequent events are occurring. And in this case, I can see them clearly under movement disorders, which includes Parkinsonism. That's where most of my events are aggregated. Now, when the need comes in for a focused search, it's a little bit different. Focused searches may be useful for deeper dives. So you already identified that maybe there's a cluster during my primary SOC output. So that's graph of the psychiatric SOC. That's my first inkling that there's a cluster here. Or maybe I already know from previous toxicology results or animal studies or known class effects that I have an identified safety concern. So I already know that I'm looking for something. I'm going to go digging for it. Or maybe I have events of special interest and I want to be very intentional about monitoring them. Or maybe I received a regulatory query and they are directing me where I need to focus my attention because they did that secondary SOC view and you didn't, and they saw something which you didn't, and now they're asking you about it. So there's different options when you're intentional about doing a focused search. And mainly the one that we're going to be showing next is the secondary SOC view. So for the method used for a focused search by secondary SOC assignment may depend on what we talked about previously, those database characteristics of the organization. Are you able to query the SOC and the HLTs and the HLGTs to include both primary and secondary SOC assignments in your data display? Can you do that without programming, or do you need some help programming? If the database does not allow automated output by secondary SOC, then can you perform the query using other resources? Do you have individual PTs in the primary and secondary SOC locations? Now, the nice thing about the MedDRA tools, the browsers, the MSSO browsers, whether it's the desktop browser, the existing web-based browser, the new web-based browser, they all provide you the ability to view options to display the secondary SOC pathways. They all allow you to use research bins and export results with the research bins using secondary SOC pathways, and they all have a tool embedded in them called the hierarchy analysis tool, which also helps you view your data with the secondary SOC. We're going to show you hopefully a demo on that next. So the benefits, of course, of using the secondary SOC is now you have a deeper dive. You have a more comprehensive view of your events of interest. But the limitations, now remember, when you're looking at secondary SOC, it means that every adverse event on your reports is showing up with all possible SOC assignments. So if it's linked to 3 SOCs, it's going to show up 3 times. That doesn't mean that your adverse events suddenly increased 3-fold. It's just you're looking at all the SOCs that are linked to that preferred term. Now, in terms of grouping terms, we already talked about that example of how to use grouping terms. The SMQs and the customized searches are topics for our next webinar. We just can't cover them here. So here is another real-world example. It's Friday, and well, it's not today, Friday, but this example happens on a Friday because in my experience, most fires start burning around 4 p.m. Friday when you're getting ready to log off. So it's Friday end of week. Most of your team has left for the weekend. You're about to sign off, and you get an urgent call from your medical reviewer. She's been in discussion with one of the PIs on the study, and she needs to know urgently, out of the early set of adverse events you've collected so far, how many cases are autoimmune diseases? And she says, get back to me as soon as you find out. So you hang up and you're wondering to yourself, well, okay, I can easily pull out my list of AEs by, primary SOC and PTs. So let's run it. And you start looking at it, and it's about 3 pages worth. So this is your first page, and you're looking at it and you're wondering, well, out of these events, how many of them represent autoimmune diseases? And you notice that the first SOC is blood, but one of the PTs says autoimmune neutropenia. And you think, okay, it has the word autoimmune in the verbatim. It's got to be an autoimmune disease. You have no clue what Evans syndrome is, so maybe it's not an autoimmune disease, and you're not sure about this platelet anisocytosis and platelet toxicity. The second SOC is cardiac, again with that word autoimmune myocarditis, which is different than standalone PT myocarditis, and you're hoping that the coders who coded these 4 cases of autoimmune myocarditis and 2 cases of myocarditis did it verbatim matching what was received, because this is another clue that if your coding is not consistent, you're not going to be aggregating events together. So you're hoping these 2 cases of standalone myocarditis don't have something in their source that may indicate they're autoimmune, because that would change your counts. But anyway, you're looking and you say, okay, so maybe so far I have the autoimmune neutropenia and the autoimmune myocarditis. So what else? You go to the next page and you see another verbatim under endocrine with the word autoimmune in the PT, and you go, okay, this polyglandular autoimmune syndrome must be an autoimmune disease, but you don't think the Burchard chorioretinopathy is autoimmune, and you don't think anything under hepatobiliary is autoimmune. And you move on to the next page and you see autoimmune disorder, but you don't think anything under musculoskeletal or skin is autoimmune. And you go, okay, everything with autoimmune in the verbatim is going to be an autoimmune disease. But is that validated? No, it's not validated, because you just based it on having a descriptor of autoimmune in the PT. That's not going to work for your medical reviewer. I don't think she's going to be happy with that output. So what else can you do? Would you go online? Would you Google them? Would you try to chase down somebody with a medical background on the team that hasn't left for the day and willing to take a look at the list? But that's not a standard approach. And besides, that person may also want to refer to a committee discussion. So the good news is that MedDRA will give you an easy way to answer this question. And that process lies in the browser. So let's jump into the browser. MedDRA has 3 browsers. It has the desktop browser, the MDB, the web-based browser, the WBB, and the mobile browser, the MMB. The mobile browser we're not going to touch today. And just as a note, it's not an app. It's just a link. It's a URL link with the interface made large enough to work on smartphone screens. So that's basically it. The desktop is a little bit different than the web-based because the desktop has all the functionalities, but it doesn't have MedDRA files. So you download it to your desktop, and then you download whatever MedDRA files you want to work with, whatever MedDRA versions in whatever language. The WBB, the web-based browser, is the one that is available online, and it's updated with every single release of MedDRA, so you don't have to download anything into it. And for our purposes, we're going to be focusing on the functionality of the web-based. But all the browsers will require MedDRA ID and password. We went over how to retrieve them. They will allow you to view all MedDRA terms and all SMQ terms, and it will enable you to run your view whatever the MedDRA language is that you want. You can even change the interface language to one you're comfortable with. And the desktop browser and web-based browser will also allow you to export any of your search results. They have this search bin that's really cool, and you can export it into an Excel file. Now, both the web-based and the desktop browser will allow you to view the secondary link information. They will allow you to upload some data and run this SMQ tool, SMQ analysis tool, which we will demo in the next webinar. And they will have the ability to code using advanced search options. We talk about these in our coding webinars. And of course, to view that pink screen, that supplemental, in the next release. But this is only for web-based, because of course, we can't upload the supplemental terms into every person's desktop browser. Once you upload the desktop browser, download it into your desktop, you're responsible to upload whatever MedDRA files you want in it. You can certainly download the supplemental and upload them into your desktop browser, but if you want that available to you online, then your best option is using the web-based browser. All right, here's my data set. So going back to my list, these are 23 preferred terms. Some of them were reported 4 times, 5 times, some had 2 cases, some had 1 case. But all in all, I had 23 unique preferred terms in my list. And my question was, which of these are autoimmune? And if I ran my listing just by primary SOC, I'll notice that under the immune system disorders, there's only one, that autoimmune disorder. I mean, it's really easy, but it's for intents of demonstration. This can't be right. So what does MedDRA offer me? MedDRA offers me 3 options. The MSSO has a nice way to answer this question by 3 options. One of the options is, why don't you put the term in to the browser and let's see what it's connected to. Let's do it now. So let's go into my browser. So let me go back to the homepage for MedDRA. There's again 2 ways to access browsers. One of them we said how to use. You go to Tools, and here's my browsers. And I have options, either the existing web-based browser or the new web-based browser. And they both have the same functionalities. Whichever one you choose, that's fine. Just bear in mind that the new web-based browser has a much stripped, neater, cleaner, simpler interface. The other way is there's a quick link. So there's a shortcut up here again, just like the other quick links, this time for WBB. And if I click on WBB, it takes me to the login page of the web-based browser, the existing one. Okay, let's go in and let's take a look at the options that we talked about. We said one of the terms, the first one, let's say the first one was anemia. I put in anemia and I hit enter. And here's my anemia. And if I look at my preferred term, here's my preferred term anemia. If I expand it, I can see the links that it's linked to, the path that it's linked to. It has one HLT, that one's linked to one HLGT, and here's my blood. So anemia is uniaxial. It has only one SOC, and I don't see anywhere that it's an autoimmune. So I can say, okay, I'm sure the anemia is not an autoimmune disease. What about the one that said autoimmune neutropenia? So autoimmune neutropenia. I don't have to spell out the full word, but I just have to make sure that the part I spell is correctly spelled, and I hit enter. Here's my autoimmune neutropenia, and let's see this path. There's my 1-to-1, 1 LLT to 1 PT. When I expand the PT, I notice 2 HLTs. One is blood and one is neutropenias, and I expand again. Here's my link to the immune system disorders. Here's my link to blood. So autoimmune neutropenia, it appeared in my listings with the primary SOC blood. It has a secondary link as an autoimmune disorders. Here's my HLGT under the immune SOC. So, okay, I'm sure now that autoimmune neutropenia is in fact an autoimmune disease. It just happened to have the primary SOC to the site of manifestation. So it manifests as a blood disorder because it's a neutropenia, but it's also linked to the immune SOC because that's the underlying etiology. Okay, so I can add this. Well, how do I add this? What do I do with this? If I right click on my preferred term, because I want to send these to my medical reviewer and say, here, I'm finding these, I can add this to something called Add to Research Bin. So what does this look like? If I click it and Add to Research Bin, it opens up this panel for me. And it tells me that you're starting a research topic. Do you want to give it a name? Well, sure, let's call it autoimmune. Okay, and do you want everything? Do you want the secondary SOCs? Well, yeah, because under primary, I'm not going to see it as an autoimmune. Well, do you want the HLTs and HLGTs? That would be nice to keep them because I see the HLGTs under autoimmune disorders. Is it enough? Well, I can export this, but does that mean I have to go through them one by one? Man, that's not going to be very efficient if I have another half hour before she leaves. So, okay, let's say no. I'm not going to export this. I'm going to try something else. I added it to my research bin. Is there another option? Let's go back to our slides and find out. So this is my first option, is that I can give it a research term. I can look for a term in the research field, and I can export whatever results it gives. Whether it's one result like autoimmune neutropenia, whether it's 50 or 100 results if I put in headache, for example. And this is the same functionality, whether I use the existing web-based browser or the new web-based browser. This is what the same thing is going to look like in the new web-based browser. So that's option one. Option two takes you a step further. It tells you, okay, you're not just looking up one term, you're looking up a bunch. Well, 23 to be specific, or somewhere around there. So can I leverage this research bin to keep going? And the answer is yes, you can add more than one to the research bin. Let's see how that's done. I added my autoimmune neutropenia. I'm looking for my next term. That's Evans. So that's Evans syndrome. Let me expand it again and look at the links. And I see two links again. One is blood, that's primary. You can see the blue box, that's a primary. And a secondary link to immune systems, again, an autoimmune disorder. So yes, Evans works also. If I right click it, I can also add this one to my research bin. So now I have two of them. Do I keep going? I mean, this is still not very efficient, and no, I shouldn't keep going. There's an easier way to do this. Let's go back and take that list and let's create an Excel sheet the way MedDRA likes it, the way the MSSO tools like it. Let's create this Excel sheet like this. Here are my 23 adverse events. These are my preferred terms. Again, with the row ID, I'm just leaving it numeric, that's fine. And here are my codes. If I'm using English, I don't need them. But this is my report, right? Column A, column B, column C. Is there a tool I can use in MedDRA? Yes, but remember to close the Excel. And let's go up here to the top. There are tools listed here, and here's your hierarchy analysis. It's a really fun tool. I mean, I highly advise you grab an output from your data, test it out. It's really fun to use. Let's see what it does. I click on the hierarchy analysis, and the first question is, what language is your data in and what version? So my data exists, let's say, in version, I don't know, 28. And I'm using the English language. I do want to keep secondary because that's part of what we're doing now, this whole thing. And HLTs, HLGTs, I don't really much care right now about the codes. Maybe, when I send this to my programmer, I might rerun it with the codes. But for now, let's see what this looks like. It's going to ask me the same things. It has to be Excel, 3 columns. I mean, we're getting used to now this nice formatting, but easy and simple that the tools want. Restricted to 100,000 because I'm in the existing browser. I'm not in the new browser. That's fine. It's going to delete the data. It's not going to store it. That's fine. Let me choose the file. Here's my secondary sock exercise. This is the one that I just created. So I'm going to double click on that. It is linked, and I'm going to click next. Again, the same thing. It's going to run the data validation. If all of them are true MedDRA data, it's not going to bother me. But if I made a mistake with the spelling with any of them, it's going to tell me row so-and-so doesn't make sense to us. We're not going to process it. But here's the output really quick. Okay, let me open it up and see what the output is. I see the 23 adverse events. So here's 23 adverse events, but I see more than 23 rows. And that's because some of these preferred terms are actually being repeated more than once. I was expecting this because I'm running the view not just with the primary sock, but with the secondary. So what are these columns? Column A is the same as the row ID that I had used. And if the same term is repeated more than once, I'm going to see it with the same row ID repeated more than once, which is fine. Here's my code. I don't really need it. Let me hide this column. The tool will tell me what level of MedDRA terms this exists. Is it an LLT or PT? Right now, that's not really an important output for me. I'm going to hide this column. The next one is, well, if you had given me LLTs, here's your PTs, but you actually gave me all them PTs. So I don't really need this one now because they're matched. They're all PTs. So I'm going to hide it. This leaves me with the HLTs, HLGTs, and SOCs, and a column that tells me which one is the primary SOC. If I filter this, and I'm only interested in autoimmune disorders, and I know there's an HLGT because I saw that when I was looking 1 or 2. If I filter this and I decide that I'm only interested in autoimmune, I wonder how many of those preferred terms are actually autoimmune disorders. I just filter and I pick. Here's my autoimmune disorders, and I go, okay. And oh my goodness, look at this. So there was only one under the immune system SOC, but there's actually 10 more that are autoimmune diseases, but they exist in MedDRA linked to the immune SOC as a secondary pathway. So imagine that you went back to your medical reviewer and said, we only have one. We only have one case of autoimmune because you only looked at your data in the primary SOC link. That is not a good day for you because you are missing the majority of them. The majority of them have a secondary link. And again, this is, of course, an example output for demonstration purposes, but you see the point, hopefully, of the importance of looking at your data in its secondary SOC. Now, I told you before, this first row is actually pretty cool because there is something you can answer specifically. You can actually give the medical reviewer the exact case IDs of all of the cases you have in your database that have any of these 11 preferred terms. Because if autoimmune neutropenia is reported 4 times and Evans is 4 times, that's 8 cases right there. But can you actually identify them? Let me show you a different way that I created my tracker. Here's the same information, only this time, instead of not using the subject ID or the case ID or the row ID, I actually leveraged it, this first column. The reason why I have more rows now than I had before is because some of these unique preferred terms actually are reported in more than one case, which makes sense. So subject 1 and subject 10, they both have anemia. Subject 1 and subject 3 both have autoimmune neutropenia, and you get it, right? So this is the purpose of creating the tracker this way. It's still a simple tracker. It's still showing me the preferred terms and the preferred codes. That's all it's showing me, only I'm adding subject IDs. And again, don't worry, the tool is not going to save your data. But how is this going to change the report that I just ran. Well, let's run the same exact thing, only use this tracker. Remember to close it. I'm going to go back to my tool. Here it is again, hierarchy analysis. I'm going to select version 28. I'm going to keep it defaulted for secondary SOC with the grouping terms, and I'm going to click next and choose my other file now, the one that has the subject IDs. Let's try it now and see what my output will look like. So let's open it up. I already know it's going to be longer than the first one because I had a longer report to start with, on top of which every single one is going to be repeated however many times is needed to show me all the SOCs associated with that PT. So again, let me just hide these 3 columns just to make it easier for the demo purposes. I don't need the code or the level or the PT because my data already exists as PT. Let me hide these. And let me introduce a filter right here. And again, I'm only interested in autoimmune disorders, but think you can actually maybe sort this alphabetically by HLGT and look for a signal or a cluster that way. So you can experiment with this and look at it from different ways and look for any safety signals that you didn't know existed. Think about what you can find if you look at it that way. But let's filter by autoimmune disorders because I need to capture the medical monitor before she leaves, and I filter. Now how many do we have? We actually have 30. We have 30 cases. These are exactly 30 cases in my database, and not only did I recognize the number that I have 30 cases, I can send her the list of which specific cases they are, and that will make her life so much easier. So I hope this was useful. I'm very cognizant of the time, so I'm going to keep going with the next item on here, and hopefully I am willing to stay on at the end if you have questions, but I'm going to try and finish the bulk of the presentation before we reach the top of the hour. Okay, so we talked about how the research bin exists in the browser. We showed that in the web-based browser. This is just a screenshot of how the same tool is available to you in the new browser. So that's just a screenshot of that. And here's the option 3 that we just used, using the hierarchy analysis tool. This is the existing browser, and this is the new browser. The same tools are available in all of them. And this is how the output shows from the existing browser of the unique preferred terms when we don't have subject IDs. And this is the same output from the new browser. So the tools are available no matter what the browser is that you're using. And when we use the subject IDs, we were able to identify the exact 30 cases. And that's from using the secondary SOC pathways. The last topic for today is how to develop queries using MedDRA. And I have a few questions, but let's make them super quick. I'm not going to stay too long. I just need to kind of prompt you. Which level of MedDRA is used for data reporting and data analysis? And we kind of touched base a little bit on these, but what do you guys think? And I'm just going to give it a few seconds. I really want to keep going in the last 5 minutes to show you how to create a query. So let's just show the responses now. PTs are correct. LLTs typically not used for reporting and analysis because they are way too granular. The other grouping terms, HLTs and HLGTs, are useful. I do hope more people are interested in looking at them. But when you're reporting to regulators, usually they are not referenced as levels of MedDRA, but PTs definitely yes. SOCs, it depends. So like we said, for example, DSUR, if you look at that guideline, you're going to see it references both PTs and SOCs as MedDRA levels to be provided. What about this question? When you're building a query, which level of MedDRA do you think would be the one to use when you're building a query? So we're just going to give it a few seconds also because I'm just cognizant of time, and I do apologize if we're going to be going a little bit over. Let's stop and see responses. PT is correct. The PT level is a very important level when you're building your query. Now, the HLTs and the HLGTs are leveraged. They are useful for you when you're selecting which PTs to include in your query. But actually, when we say query, we say the list of terms that you're going to be cross-checking against your data. So PTs are correct. LLTs, not typically used when you're building your own query because they become kind of harder to manage to ensure that you have all of them, and if some of them change and move to different PTs and different version. So the approach is generally build your query at the PT level. That's the answer. Well, what's a query? What do you mean by query? Query is a list of preferred terms that are related to each other to cover a topic of importance to you. This topic of importance may be something as detailed as blood pressure decreased, or it may be something as wide as renal disorder or liver disorder. So, whatever the topic of interest to you is, that's your safety question. And you're trying to collect preferred terms from every part of MedDRA that you think belong together to answer that safety question. Because what you're doing is once you have that collection of preferred terms, and of course, all of these preferred terms have their subsumed LLTs as part of them, but you're building the query at the PT level. Once you have that list, you're going to run that list against your database, whether it's a safety database or a clinical trial database or whatever kind of database that you have. You're going to compare it against this list of PTs that you collected and see if you get hits. That's basically a query. So your first step in any query is, what are you looking for? If you answer that question, but it's not as easy as it seems from first, because if you include way too many vague symptoms, if you include things like pain or maybe headache, depending on your condition, some of the signs and symptoms may be so vague and non-specific that you're going to be way too, like you're casting your net way too wide. You're going to get too much noise. It may be prompting you to review hundreds and hundreds of cases. At the flip side of it, if you don't include certain things, you're not going to find them. So any PTs you exclude, you're not going to be looking for them. So you have to be careful what your inclusion and exclusion criteria are. And you do need folks who represent different functional groups. It's good to have folks who understand MedDRA, of course, because you're building a query in MedDRA. So they're going to help you find terms in MedDRA. But it's important to have understanding of maybe medical background, medical conditions, how diseases manifest, what are their names, what are their diagnoses, what are their syndromes, what are their signs and symptoms, what kind of blood work would you look for, any elevated enzymes, any x-rays that are abnormal specific to that condition. Maybe if somebody had a surgery, that would be highly indicative that they had something going on, like we saw in kidney transplant. Again, what are the kind of risk factors? So you're thinking about those three uniaxial conditions. So it is very important when you're building your query that you take your time and you understand what you're going to be including and what you're not going to be including. One of the things that's nice to look are neighbors, and neighbors are found this way. So let me show you an example of a neighbor. Let's say I was interested in Evans syndrome. Let's stay on here since we have it already. How do I find the neighbors of Evans syndrome? I right click on it and I choose something called Go to Browser. Because basically what I'm telling the browser, show me Evans syndrome, the way it resides, where it lives in the browser. I want to see the neighbors. Now, when I right click on it, it's going to tell me, well, are you interested in seeing it as a immune disorder or a blood disorder? So if my interests are autoimmune disorders, I wanted to see it as an immune disorder. So I click on it here, and it's going to show me exactly the location of this PT, Evans syndrome, within a bunch of neighbors under the HLT blood autoimmune disorders. So if I'm interested in blood autoimmune disorders for my query, maybe I want to take a look at all of these different PTs and decide if I want to add them. to my query that I'm building. So that's what we mean by neighbors. And we said for LLTs, they may be a bit too granular for your query, but there may be exceptions to that rule. So typically, for example, in infections, we have the PT levels may tell you the pathogen, like this is a bacterial infection, or something similar to that, like this is an Eromonas infection. But if you're interested in the species, that level of detail, you'll find it at the LLT level. So just something to bear in mind if infections are an area of interest for you. Okay, how do we actually document what we did for the query? We have to first save the process, store the query the way we created it, and document what we did to get there. Document our definition of the condition, our inclusion guidelines, exclusion guidelines, or criteria, and then what we plan on doing to maintain it. Because remember, every 6 months, there's going to be new PTs added to MedDRA. Could any of them be useful for you? So that's an important reason to look at that version report. Or maybe PTs that you already used have now been demoted in the next version of MedDRA, or maybe not even demoted, but also demoted and then made non-current. So it's important to look at the changes that happen to MedDRA and maintain your query that you built and document everything. So here's an example. You're interested in cardiac arrhythmia. A good place to start is the HLGT cardiac arrhythmia. So let's find that HLGT. When you look for anything in the browser, it gives you different levels of terms in MedDRA that match your search. So cardiac arrhythmias are found at the LLT level, PT level, HLT level, and HLGT level. So if I right click on it and go to browser, it's going to show me that HLGT in the browser under the cardiac SOC, and I can expand it and decide if I'm interested in all the kinds of cardiac arrhythmia, or maybe one specific one like the supraventricular. And I can review these PTs either with my physician or my medical coder and folks who are interested, and make a decision if all of these PTs are of interest to me and I want to add them to my query. But how do I add them? Do I copy them one by one? No, the browser will help you copy them all. If you right click, so again, the right click is really useful. The options are really useful. If you right click, you can add not the HLGT. I don't want to look for the HLGT. I want the PTs within that HLGT. So I want to select the level to be added to my research bin. But before that, I've already used my research bin. Let me make sure I clear it because I don't want any of these autoimmune diseases in here. So I'm going to clear all, and then I'm going to close it, and then I'm going to start a new research bin for this particular topic. So I'm going to right click and add the selected levels, and I just want the PT. We just said I'm just interested in the PT. When I click next, oh my goodness, look at this. I already have a query that has all of these PTs that represent cardiac arrhythmias. This is great. But what else can I look for? Is this enough? No, it's not enough. I got to go back and I got to make sure I look in those uniaxial system organ classes. I got to take a look at the investigations, maybe cardiac investigations, specifically maybe heart rate investigations. And maybe I like the one that says, I don't know, heart rate irregular. Maybe I like this one. I right click it, and because it is already at the PT level, I'm just going to add it to my research bin. So I didn't lose any of the previous ones. I'm just going to keep adding to them. So you can see the concept here. I go through the investigations. I pick the ones I like. Now remember, with investigations, you don't want to really pick the entire group because investigations will have normal, abnormal, increased, decreased, just the fact that it was done. So it really will introduce a lot of noise if you introduce groupings from the investigation SOC. What about the surgical SOC? If I look at surgical and I find the cardiac group, and under the cardiac, maybe I'm interested in devices because a lot of these folks end up with either pacemakers or defibrillators, and I pick this one and I say, I like this preferred term. I'm going to add that one to my research bin. So now my research bin is growing, but there's something that is an important reminder, not just about going up, down with the HLGT, not just about picking terms that have arrhythmias, and not just about non-multi-axial socks. There's something very important. When you're looking at, and this is example of investigations, sorry, and this is in example of the surgical and procedure terms that may be relevant to your query. This is the one that I want folks to remember. Signs and symptoms of these cardiac arrhythmias are not necessarily going to all be under the cardiac sock. Some of them manifest as different system organ classes. So if you have a dizziness, if you have patient complaining of dizziness, that's not a cardiac sock. If you have shortness of breath, that's respiratory, that's not a cardiac sock. Some people may even present their first presentation, maybe that they had a sudden death. And believe it or not, fatal events and death in MedDRA are under the general system organ class because causes of death could be unknown. It could be multiple diseases. So it makes sense that it's under the general disorder SOC. But don't forget that just because you're looking for a cardiac condition doesn't mean you ignore all the other system organ classes. And that's where it's important to have feedback from different members of different backgrounds and expertise when you're building your query. So how do I use the query once I built it? Well, this is an example of a query on cardiac arrhythmia that a medical monitor reviewed. And when you see this example of the assessment of the medical monitor, most of them are potential cases. I mean, the unlikely ones, nausea, vomiting, makes sense. Breast cancer makes sense. This one, cerebrovascular accident, you may or may not agree with the medical monitor, but he marked the rest as potential cases, even though when you're looking at the primary SOC, many of these are not even cardiac. So the sudden death is relevant, but it's under the general SOC. The ECG abnormal is relevant, is under the investigations. Dyspnea under the respiratory. Syncope under the nervous system. Cardioversion is a relevant surgery. If you're looking for congenital, the QT syndrome, that's a congenital SOC. And look at this. This is a risk factor that the subject already had, an existing cardiac assistance device. He was an existing cardiac assistance device user. That's social circumstances. So it's very important to remember that just because you're interested in cardiac diseases or conditions, don't limit yourself to the cardiac SOC. Look through MedDRA. It is definitely worth it. You can apply the same approach no matter what the topic of interest that you're using. So how are these then exported? So if you show your research bin, let's say just for the points of this demonstration, I did all that and I looked through the entirety of all the system organ classes and I'm happy with this list. I want to send it to my programmer now and have him run it through my database. I can export it. And if I export this list, it will open up as an Excel sheet. And I'm at liberty to save it, rename it, keep all the different hierarchies, or just send him the list of PTs and say, please run it against this PT. And if I want to send it to an IT folk colleague, I can, include that tick box for the codes now, may be useful, and send him the PTs as well as the codes. But you get the picture now. It's easy to export this. It's easy to start looking at this. And if I had selected this to show me multiples and in primary and secondary, it's going to show me all of them. If I'm just interested in the primary SOC, because this is not for analysis, this is just for testing my data, then maybe I don't need the secondary SOC. I just need the primary. So this is a way to create a query and use a query. And just as a reminder that the tools provided by MedDRA are really good, strong, powerful tools for your data review and analysis. But be patient. It does require being more knowledgeable with MedDRA, like being trained with these webinars, and it does require help from folks with medical knowledge and medical background, but it's well worth it. It's well worth the effort when you're developing and sharing queries. And just as a teaser for the next webinar, the browser already has built-in queries that were built very similarly to the process we just went through. If you change the browser view from a SOC view to an SMQ, you're going to see a long list of built-in queries that MedDRA already created for you, covering multiple topics. And if your question, well, is cardiac arrhythmias there? Yes, it is. And it has multiple sub SMQs, and you can pick and choose whichever kind of cardiac arrhythmia you're interested in, or run all of them. And that's something we're going to cover in the next webinar on how to run and leverage these built-in queries and this built-in SMQ analysis tool. So we summarize here by saying that we presented you with the MedDRA data retrieval and presentation points to consider documents. We discussed applications of MedDRA and went over different demos of the different tools. The slides also offer you some of the contact details, where to access the browser if you have questions to our help desk. And of course, if you want to sign up for more training. So Hannah, I'm going to stop here and open it for Q&A. I'm willing to stay on if folks have questions, but just a reminder to folks who cannot stay with us, if you do have a question, please feel free to reach out to our help desk, and they will either help you directly or forward it to me. So any questions, Hannah?
Our first question asks, could you please provide an estimation or share your insight of the percentage of trial databases currently organizing/keeping study data using only primary SOCs compared to those utilizing both primary and secondary SOCs? Is there a prevalence known to MedDRA experts across the industry as of today?
So that's an interesting question, but I don't have a number to give to you. Now, the reporting, when you report your data, the expectation is that when you report the data, it will be primary SOC data, because you don't want to cause confusions or any questions about your frequency, your adverse event profiles and your adverse event frequencies. However, your leveraging of the secondary SOC information is very useful because it will cover details of the view of the data that was not clear to you in the primary SOC. So if you want to be very prudent, my advice is use your secondary SOC, leverage it, make sure you understand the safety profile of your products and that there's nothing hidden in there. Because when you send your data to the regulator, they just need the PTs, and they'll be able to run the assessment by primary SOC, by secondary SOC. They don't need you to submit that for them. So they just need the PTs from you, and they'll be able to leverage the tools that are provided, whether in the browser or if they have their own. So whether or not you submit your data showing secondary is not the issue here. The issue here is, do you understand the safety profile of your products? How are you leveraging all the possible views that you can look at your products? Because believe me, regulators definitely are. And it's going to be a little bit of a stressful time if you get questions from the regulator because they checked the secondary analysis and you did not. Okay, any other questions?
Our next question asks, when I want to retrieve complete list of OPRI non-AE terms from a specific MedDRA version, is it possible to apply data validation or filtering to restrict results to terms present across multiple hierarchies?
So you can restrict the version right off the bat in the browser. So the browser goes down to 2.1, and you can certainly open new browser windows, and each browser window can have a different version. So whatever the MedDRA versions that you're using, the tools that are available do allow you the freedom to navigate changing the MedDRA version. And when you're running the tools, like we showed you, the hierarchy analysis tools, the MVAT tools, they always give you the option of changing the MedDRA versions. So I hope that was the main question on your end, is that yes, you can restrict your analysis based on the MedDRA version, but I'm not sure if that was really your question. So if something else that we missed, please come back and explain the question a little bit further.
Wonderful. Our last question at the moment asks, could you please let me know if a fictional data set is available on the MedDRA website for self-training purposes?
No, we don't have a fictional set of data. I think the face-to-face training is an opportunity to receive that kind of information, but it's not really that hard to create your own fictional set of data. And even if you use real data that you trim maybe selectively from your organization, it's safe to use them on the tools that I showed you, because the data is never saved in any of these tools. In fact, if you don't export it, you lose it. So unfortunately, no, we don't have that kind of listings available.
That looks
Any
like
other
our last
questions?
question for the session.
All right. Okay. Well, I do apologize for running over, and I do appreciate everybody hanging around to the bitter end. But I do hope that you found it useful, and I do hope that you come away with the sense that MedDRA really is created with the design to help you. It's really created with all of these ins and outs, with that purpose of making it clearer for you to help you understand the safety profile of your product. So please do consider leveraging the different hierarchies in MedDRA and the different tools available by the MSSO. And please come back with feedback if you have any once you've had that chance. Again, join us in 2 weeks. We're going to go over the SMQs and more about customized queries. Until then, thank you so much. Have a great rest of your day and a wonderful weekend ahead.